Today we are talking about a scientist who truly needs no introduction, but we're going to give her one anyway.
You must have to.
Dr. Jennifer Doudna, Nobel laureate, co-discoverer of CRISPR. I mean, her name is basically synonymous with rewriting the code of life itself.
It really is. It's hard to overstate the shift she helped engineer. Her work at UC Berkeley gave us this incredible power to edit DNA with surgical precision. Humans, plants, animals, you name it.
And for about a decade, that's where the story has been, right? Using this amazing tool to fix diseases inside our own cells.
Right. The focus has been very internal.
But here's the pivot. Here's the part that, signals that Doudna has the guts to take on a challenge that is just orders of magnitude bigger.
I see what you did there.
Because she and her colleagues are now aiming that precision tool outside our cells. They're targeting the microbiome.
That's the crucial turn. They are literally redirecting this molecular machine away from our own genome, our 23,000 genes, and pointing it at the trillions of microbial genes that live in us and all around us.
So this is not just about curing rare genetic diseases anymore.
Not at all. This is about engineering entire ecosystems to solve huge global problems.
Yeah.
We're talking antimicrobial resistance, immune disorders, even climate change.
Okay, so that's our mission for today. We're going to trace this whole journey. We'll start with the origin story, that aha moment.
Right.
Then we'll get into the clinical wins, the real-world cures that are happening right now. We'll tackle the biggest hurdle they face, which is delivery.
The delivery problem is everything.
And then finally, we will dive into this truly audacious new frontier, programming the microbial world. We're going from editing a single human cell to engineering the planet.
It sounds like science fiction, but it's happening.
So let's start at the beginning, because the origin story of something so high-tech is, well, surprisingly low-tech.
It really is. It's a quintessential Berkeley story. Doudna talks about this a lot. The whole thing started about 15 years ago, not in some sterile high-security lab, but over a chat at the Free Speech Movement Cafe on campus.
That's amazing, just a coffee and a conversation.
Exactly. It's that kind of serendipity, that fuels discovery. A colleague was just talking about this weird thing they'd observed in bacteria.
Which was?
An immune system, an adaptive immune system.
Hold on. Bacteria have immune systems. I was thinking of, white blood cells and antibodies, this huge, complex thing in mammals.
And that was the mystery. How does a single-celled organism remember an attacker and fight it off later?
So what's attacking them?
Viruses. Specifically, viruses called bacteriophages, or just phages for short. They're everywhere, and they only prey on bacteria.
So Doudna's lab just dove into that question. How do they fight these things off?
They did. And what they found was it was beautiful, so elegant. When a phage attacks a bacterium and the bacterium survives, it learns.
It learns.
It literally learns. It has this pathway that lets it snip out a tiny piece of the invading virus's DNA.
Like a souvenir.
Kind of like a molecular mugshot, and it stores that little piece of DNA in its own genome in a special place.
And that place is CRISPR.
That's CRISPR, clustered, regularly interspaced, short palindromic repeats. It's just a fancy name for this genetic filing cabinet, this memory bank of past infections.
So it's got the wanted posters. How does it use them when the bad guy shows up again?
This is the genius part. If that same virus attacks again, the bacteria quickly makes an RNA copy of that stored mugshot.
Okay.
That little piece of RNA then acts like a GPS tracker. It floats around until it finds a perfect match.
To the new invading virus's DNA.
Exactly. And it's attached to this other molecule, a protein called Cas9.
A molecular scissors.
The molecular scissors. The RNA guide leads the Cas9 scissors directly to the target, and once it locks on, snip.
It just shreds the viral DNA.
It shreds the viral DNA. The threat is neutralized. It's a targeted, programmable, adaptive immune system.
Wow. Okay, so that's amazing for bacteria. But how did Doudna and her collaborator, Emmanuel Charpentier, see that and think, human gene editor? That feels like a huge leap.
That was the Nobel-winning insight. That was the aha moment.
Right.
They looked at this system, the RNA guide and the Cas9 scissors, and they asked a really simple but profound question.
Which was?
If the bacteria can program this system to find any viral DNA just by changing the RNA guide, why couldn't we make our own RNA guide to target any DNA sequence we want in any cell we want?
Yeah.
A plant, an animal, a human cell. The Cas9 enzyme doesn't care whose DNA it is. It
Yeah.
just follows the guide RNA.
So you can hijack the whole system.
You can hijack the entire system, and suddenly you have a programmable word processor for genomes.
That paper came out in 2012, right? And
Right.
it just exploded after that.
Immediately. Because the power is in that programmability.
Right.
Before CRISPR, gene editing was like trying to fix a typo in a giant library with a sledgehammer. It was clumsy, incredibly expensive, and slow.
And CRISPR was like, find and replace.
Exactly, find and replace. Suddenly it was cheap, fast, and it worked in basically any cell type.
That must have created a research frenzy. I mean, how do you even keep up?
You can't. I mean, dozens of papers were coming out every week, new Cas enzymes, it's not just Cas9 anymore, new ways to use it, new strategies.
So what did they do?
Donna's team at the Innovative Genomics Institute, or IGI, they built a website called Caspedia.
Caspedia.
Yeah, it's basically a living catalog, constantly updated by volunteers around the world, just to keep track of the explosion in the CRISPR toolbox. It's a field that's evolving in real time.
Okay, so let's move from that cafe discovery to the clinic, because this isn't theory anymore. In December 2023, the FDA approved the very first CRISPR-based therapy.
The UK approved it too. This is the moment it becomes real for people.
This is the payoff.
This is the payoff. And that first therapy targets sickle cell disease, SCD. It's really the perfect proof of concept. A terrible, devastating disease caused by a single typo in a single gene.
And that one typo makes red blood cells.
Yeah.
What, they become rigid, sickle-shaped?
Exactly. They get stuck in blood vessels, which causes these episodes of just excruciating pain, organ damage, strokes. It's a brutal, lifelong condition.
So how does CRISPR fix it? Does it go in and correct that one typo?
You'd think so, but it actually does something much, much cleverer. It uses a kind of biological workaround.
Okay, tell me more.
So when we're in the womb and as babies, we don't use the adult hemoglobin that sickle cell affects. We use a different kind, fetal hemoglobin.
Right.
After birth, our bodies flip a switch. It turns the fetal hemoglobin gene off and turns the adult one on.
And in SCD patients, that adult version is the broken one.
That's the broken one. So the therapy doesn't even bother trying to fix the broken adult gene. That's a complicated repair job.
So what does it do?
It just uses Cas9 to go in and snip the off switch for the fetal gene. It turns fetal hemoglobin production back on.
So you start making the healthy baby version of hemoglobin again as an adult.
Precisely.
Yeah.
And fetal hemoglobin works great. It carries oxygen perfectly, and it doesn't cause sickling. If you can make enough of it, it completely overfeeds the symptoms of the disease. It's a functional cure.
Wow. And the sources point to the story of Victoria Gray. She was the first patient in the U.S. to get this.
Her story is, it's just incredible. She received the treatment almost 6 years ago now.
Yeah.
Her life, her own words, is completely changed for the better. The pain crises, the hospital visits, gone. The symptoms haven't come back. It's a one-time treatment that gave her her life back.
That's the dream of this technology right there.
It absolutely is.
So sickle cell's the big win. But they're obviously looking at other targets. What about something like Huntington's disease? That's another brutal single gene disorder.
It is, and conceptually the editing part is similar. You know what you need to fix.
What?
But the problem, the massive, massive problem is delivery.
Getting the CRISPR machinery to the right place?
Exactly. With sickle cell, you can take cells out of the body to edit them. With Huntington's, the target cells are neurons deep inside the brain.
And you can't just take those out.
You can't take those out. And they're notoriously hard to get things into. So the entire challenge for Huntington's right now isn't the cutting, it's the shipping. How do you get the molecular scissors safely to the right address in the brain?
So the tool is perfect, but the delivery truck is missing.
That's a great way to put it. Solving that delivery puzzle is the whole ballgame for neurodegenerative diseases.
And what about moving beyond just curing disease to prevention? I read about using CRISPR for things like high cholesterol.
Yes, this is the next frontier, the prophylactic use. We're seeing commercial companies developing therapies that would be a one-time injection.
For cholesterol.
For cholesterol, it would go into your liver cells and edit the genes that produce molecules responsible for high cholesterol, like PCSK9.
So instead of taking a statin pill every day for the rest of your life,
You get a single shot that permanently lowers your risk of heart disease.
that would be a paradigm shift for public health.
It would be the first widespread protective use of gene editing, moving it from rare diseases to one of the biggest killers on the planet.
Okay, so let's really dig into this delivery problem, because you said it's everything. The approved sickle cell therapy works, but the process itself sounds grueling.
It is. It's grueling, it's incredibly expensive, and it limits who can get it.
Because it's an ex vivo therapy. Right.
Yeah.
Outside the body.
Exactly. Let's walk through it. First, they have to harvest bone marrow stem cells from the patient.
Okay.
Then, those cells go to a highly specialized lab where they're edited with CRISPR.
And while that's happening,
The patient is in the hospital undergoing high-dose chemotherapy
why?
to completely wipe out their existing unedited bone marrow to make space for the new corrected cells.
Wow. So you're talking long hospital stays, huge risks, and it must cost a fortune.
Hundreds of thousands, sometimes millions of dollars per patient. It's just not scalable. It's not a solution for the world.
So the holy grail is in vivo editing, getting the CRISPR machinery delivered directly into the patient's body.
That's the dream. A simple injection that goes to the right cells and does the editing on site.
And to figure out how to do that, they turn to a virus.
It's wonderfully ironic, isn't it? They looked at the one thing in nature that has perfected the art of getting inside our cells.
Viruses are master delivery vehicles.
The best there is. They've evolved for billions of years to be incredibly efficient at targeting specific cells. A flu virus knows how to find lung cells. Other viruses know how to find liver cells or brain cells.
So the challenge is, how do you use the virus's delivery system without its, disease-causing payload?
Exactly. You want the envelope, but not the letter inside. And that's precisely what Doudna's lab did. Work led by a former postdoc, Jenny Hamilton, developed something they call enveloped delivery vehicles, EDVs.
EDVs, so what are they?
They're essentially molecular Trojan horses. They take a virus, strip out all of its viral genes, the infectious parts.
So it's an empty shell.
A completely empty, non-infectious shell, and then they pack it with the good stuff, the Cas9 protein and its guide RNA.
So the payload is now the gene editor. But how does it know where to go? How do you program the address?
That's the other brilliant innovation. The surface of the EDV is programmable.
What does that mean?
It has two key things on its surface. First, molecules called fusogens, which help it merge with the target cell membrane, just like a real virus would.
So that's the key to get inside.
That's the key. And the second thing is the address label. They decorate the surface with tiny fragments of an antibody.
An antibody.
Yes. And you can choose an antibody that specifically recognizes and sticks to only one type of cell. A T cell, a lung cell, a brain cell.
So you can change the antibody fragment to change the target organ.
Precisely. You use the same Cas9 payload, the same viral shell, and you just swap out the antibody to send it anywhere you want in the body. It makes it incredibly flexible and scalable.
And did it work? What was the proof of concept?
It worked beautifully in mice. They gave humanized mice a single injection of these EDVs that were programmed to target human T cells, and they saw precision genome editing only in the T cells, nowhere else. It was surgically precise.
That's incredible. So this is the path to making gene therapy a simple shot.
It's the path to making it safer, much, much cheaper, and accessible to everyone on the planet, not just people who can afford $1 million hospital stay. It's a total game changer for medicine.
And this delivery tech is obviously going to revolutionize human medicine. But as we said at the top, this is where Doudna and her team take this quantum leap.
Yes.
Moving beyond the human genome to the trillions of other genomes living on us and in us. The microbiome.
It's such a radical shift in thinking. The human genome is a blueprint, but the microbiome is a dynamic living ecosystem.
Right.
And if you can program the genes of the microbes in that ecosystem, you can engineer the entire system's output.
Okay, let's start with the most surprising application you mentioned. Climate change. The partnership with UC Davis to edit the microbes inside cows.
Yeah, this one is it's mind-bendingly ambitious.
What's the problem they're trying to solve?
Methane. A huge portion of global methane emissions, which is a super potent greenhouse gas, comes from livestock, from cattle.
And that's not from the cow itself, it's from microbes in its gut.
Exactly. In its first stomach, the rumen. There are microbes in there called methanogens, and their whole job is to produce methane as a waste product.
So the CRISPR solution isn't to edit the cow.
Nope. It's to edit the microbes inside the cow. The idea is to go in and use CRISPR to just tune down the genes responsible for making methane in those gut microbes.
You're giving the cow's gut flora a software update.
That's a perfect analogy, and it might even have an economic benefit.
How so?
Well, making methane wastes energy. If those microbes aren't wasting energy making methane, that energy could theoretically go back to the cow.
To make more milk or more meat.
Potentially. So you have an environmental win and a commercial win all in one. It's systems engineering on a planetary scale.
Okay, so that's the environment. Let's bring it back to human health. Applying CRISPR to our own gut microbiome. The sources lay out three main strategies.
Great. You can think of it as additive, subtractive, and modulatory.
Let's start with additive. What does that mean?
Additive therapy is about engineering a friendly microbe to act as a living drug factory inside your gut.
A designer probiotic.
Exactly. You take a safe, well-studied probiotic, like a strain of E. coli called Nissel 1917, and you give it a new job.
Like what?
For something like inflammatory bowel disease, IBD, you could engineer it to constantly produce a powerful anti-inflammatory molecule right at the site of inflammation.
So instead of a systemic drug that affects your whole body.
You have a gut resident making the medicine exactly where it's needed. They're also engineering them to break down toxic compounds, like for the disease PKU, which we can get into.
Okay, so that's adding a function. What about subtractive therapy?
Subtractive is all about targeted killing. This is the strategy aimed squarely at the crisis of antibiotic resistance.
So this is like a sniper rifle instead of a bomb.
A perfect analogy. Instead of broad-spectrum antibiotics that wipe out your whole gut ecosystem, good and bad,
Right.
you use a CRISPR phage system. You take one of those bacteria-killing viruses, a phage, and you arm it with CRISPR.
And you program the CRISPR to, what, find the bad bacteria?
You program it to find a DNA sequence that is unique to the harmful antibiotic-resistant strain. When the phage injects the CRISPR system, it finds that sequence and just shreds the bacteria's own genome.
It's turning the bacteria's own defense system against itself.
It is. It kills the bad guy and leaves all the beneficial neighbors untouched. It's precision antimicrobial medicine.
Okay, additive, subtractive, and the last one was modulatory.
Modulatory is the most subtle. It's not about adding or killing, it's about fine-tuning.
How do you do that without cutting the DNA?
You use a version of Cas9 that's been deactivated. It's often called deCas9 or dead Cas9.
Dead Cas9.
Yeah, it's been engineered so it can't cut DNA anymore, but it still has its guide RNA, so it can still find any gene you want.
So what does it do when it finds it?
It just sits there. It binds to the DNA and acts like a big roadblock, physically blocking the cell from reading that gene.
So it's a dimmer switch.
Huh?
Or an off switch.
Exactly. A temporary reversible off switch. You can dial down a gene's expression without permanently breaking it. This is huge for studying what genes do, but also for therapeutics, where you might want to just gently tweak a microbe's metabolism instead of killing it.
That subtractive therapy for antibiotic resistance sounds incredibly important. Let's drill down on that. The sources talk about a specific strategy called conjugative probiotics, or COPS.
Right. The COP strategy is a really clever piece of engineering because the gut is a tough place to work. It's dense, it's competitive.
So just throwing in your killer probiotic isn't enough.
It's not. You need your therapeutic weapon, the CRISPR killing module, to spread efficiently from your engineered probiotic to the dangerous bacteria you want to eliminate.
So how do you make it spread?
You hijack the way bacteria naturally share genes. It's a process called conjugation.
This is like bacterial sex, right?
Basically, yeah. One bacterium builds a little bridge to another and passes over a piece of circular DNA called a plasmid. This is how antibiotic resistance spreads so fast in the first place.
So the researchers weaponized that process.
They did. They took that friendly E. coli Nissel probiotic and gave it a special plasmid that's been evolved to be incredibly good at transferring itself. And on that plasmid, they loaded the CRISPR-Cas9 killing module.
So the friendly probiotic becomes a courier, delivering a poison pill to the resistant bacteria.
That's it. And the key was really optimizing that plasmid transfer rate. That was the bottleneck before. They made it super efficient, even in the complex environment of a real gut.
So, did it work in a real gut? In
Yeah.
a mouse model?
It works spectacularly. In mice, a single dose of their engineered EB Cox strain eliminated over 99.9% of the targeted resistant E. coli.
99.9%.
Huge drop. And then they tested it against an actual infection with a pathogen called Citrobacter rodentium. After 4 days of treatment, full clearance, the infection was gone.
Wow. That suggests a future where you treat a nasty gut infection, not with antibiotics, but with a dose of another smarter bacteria.
A living drug that seeks and destroys only the pathogen. It's huge proof of principle.
But there's always a but, isn't there? The promise in the lab doesn't always work out in humans. And the sources point to the phenylketonuria or PKU trial as a really important reality check.
The PKU story is essential. It's a lesson in humility for the whole field.
So remind us what PKU is.
It's a genetic disorder where you can't break down an amino acid called phenylalanine, or Phe. It builds up and causes severe brain damage if you don't follow an incredibly restrictive diet your whole life.
So the idea was an additive therapy, right? an engineered microbe to break down phe for you.
Exactly. They engineered that same E. coli Nissl strain to gobble up phenylalanine in the gut before it gets absorbed.
And the early trials looked good.
They looked promising. They showed it was safe, and they showed the bacteria were actually doing their job. They were metabolizing feed. They could measure the byproducts. The core concept worked.
So why was the big phase 3 trial discontinued?
This is where the beautiful, clean world of the lab crashes into the messy reality of the human body. It wasn't a safety issue. It was an efficacy issue. It just didn't work well enough.
Why not?
Two huge reasons: interpatient variability and, crucially, a failure of engraftment.
Engraftment, meaning the new bacteria didn't stick around.
Exactly. They would colonize for a little while in small amounts, but then they'd get flushed out. They couldn't set up a stable, long-term residence in the gut.
And without that, they can't provide a consistent therapeutic effect.
Right. And every patient is different. Their diet, their existing microbiome, their medications, the variability was just too high. The engineered microbe worked, but it couldn't reliably live and work at a high enough level in the human gut to make a clinical difference.
That's such a critical lesson. It's not enough to build the perfect microbe. You have to figure out how to make it a permanent resident.
It's a massive focus for the field now. The biology of colonization is just as important as the synthetic biology of the therapy itself.
And to tackle these complex problems, they're using more than just the original Cas9, right? The toolbox is expanding.
Oh, absolutely. Cas9 is the famous one, but for the microbiome, you need different tools for different jobs and different types of bacteria.
Like what? The sources mentioned Cas12a.
Cas12a, or CPF1, is a real workhorse for editing some of the most common and important gut bacteria, like bacteroids. It's much more efficient than Cas9 in those species for making big changes, like deleting an entire gene pathway or inserting a large piece of new DNA.
So it's a heavy-duty tool.
A very heavy-duty tool. You see editing rates over 60, sometimes even 100% with Cas12a in bacteroids, which is just incredible.
And then there's Cas13, which is totally different.
Totally different. Cas13 doesn't touch DNA at all. It's an RNA-guided RNase. It cuts RNA.
So why would you want to do that?
Because it gives you that dimmer switch we talked about, but in a different way. RNA is the temporary message from a gene. If you cut the message, the protein never gets made, but you haven't permanently altered the DNA blueprint.
So it's a way to temporarily knock down a gene's function.
Exactly. You can turn off a toxin, fight an RNA virus, or just study what a gene does by turning it off for a while. It adds another layer of control to the toolbox.
So we've gone from the origin to the clinic to this wild new frontier of microbial editing. Before we wrap up, it's worth just taking a second to appreciate the sheer scope of where CRISPR is in the clinical world right now.
It's staggering, really. There are dozens and dozens of clinical trials happening right now all over the world, phase 1, 2, and 3.
And the main focus is still on blood disorders.
That's where the initial big successes are. So yes, a lot of work in blood cancers, hematological malignancies, and the hemoglobinopathies like sickle cell and beta thalassemia.
But it's branching out.
Oh, absolutely. It's moving into metabolic disorders like type 1 diabetes. They're targeting chronic viral diseases like hepatitis B and HIV. It's clear that this isn't a niche technology anymore. It's becoming a core pillar of medicine.
And with that kind of power comes a lot of responsibility. Dowda has been very vocal about the ethics from day one, hasn't she?
She has, and it's one of the most important parts of her legacy, I think. She recognized the risk immediately. Back in 2015, she helped organize the first big international summit to talk about the responsible use of CRISPR.
And the big scary topic was germline editing, right? Editing embryos.
That was a central concern, making changes that could be passed down through generations, creating, designer babies.
And that meeting really set the tone for caution and public dialogue.
It did. It established this precedent that the scientists have to lead the ethical conversation, not just the scientific one. It's about moving from what can we do to what should we do.
So looking forward then, what is the ultimate goal? Where does this all lead?
The long-term vision is it's to go beyond these single-gene diseases, as important as they are.
To the really complex stuff.
To the really complex multi-gene diseases that affect most of us, Alzheimer's, heart disease, most cancers. That requires solving all these delivery problems we've talked about and learning how to regulate dozens of genes at once, very subtly.
So the vision is a future where a one-time genetic intervention could treat or even prevent conditions that today require a lifetime of pills and procedures.
That's the dream. To make gene therapy a safe, accessible, standard of care option for everyone.
So we started this deep dive with Jennifer Doudna, and a discovery that let us edit a single gene inside a single human cell.
Right.
We saw that lead to a cure for a disease like sickle cell. We saw the massive challenge of delivery and the ingenious solutions being developed.
EDVs, yeah.
But the real takeaway today seems to be that the biggest revolution is actually happening outside of us, in this world of microbes that shapes our health and the health of the entire planet.
It's a profound shift. And that lesson from the PKU trial, that you have to master the host environment, not just the molecule. That's the key. That's what they're applying to everything now, from IBD in the gut to methane in a cow.
It's no longer just about editing the human genome. It's about engineering nature's operating system.
It really is.
So we'll leave you with this final thought to pay on. If they can successfully tune the microbes inside a cow's stomach to fight climate change, what other huge planetary-scale problems could be solved by rewriting the code of the microscopic world?
Think about it. Programmable microbes that eat plastic in the oceans. Engineered soil bacteria that let us grow food in the desert. The technology that started with a casual chat in a cafe is now positioned to solve some of the biggest challenges we face. The revolution really has found its guts.
That is our deep dive for today. Thanks for joining us. We'll see you next time.